mRNA leaves the nucleus into the cytosol where it is used to make protein in the process of translation. mRNA is in the cytoplasm and binds to a free ribosome, which reads the information to link together amino acids to make the polypeptide. The newly synthesized polypeptide has information in the N-terminus that signals for it to be carried to the rough endoplasmic reticulum. This sequence is called the ER signal sequence. Signal recognition particles recognize the signal sequence and binds to both the signal sequence and the ribosome, arresting further translation. SRP ribosome nascent polypeptides bind to the cytosolic surface of the ER membrane. The SRP and SRP receptor bind together, and the ribosome and translocon bind together. SRP is then released from its ER receptor, and the ribosome becomes attached to the translocon. Signal sequence on the nascent polypeptide is inserted into a narrow aqueous channel of a translocon, displacing the plug within the translocon. The growing polypeptide is translocated through the hydrophobic pore ring and into the ER lumen. Translation is complete when signal peptidase cleaves off the signal sequence. Membrane-bound ribosomes is released from the ER membrane, and the plug is reinserted into the translocon channel. As soon as asparaginine is available, oligosacchαρyltransferase takes the oligosaccharide from the dolichosyl phosphate and transfers it to the polypeptide, where it is attached to an asparaginine, which can occur before the polypeptide is fully released into the ER lumen. Two glucose molecules are cleaved off. And the ER chaperone, calnexin, binds to the last glucose molecule and works with disulfide isomerase to make sure the polypeptide is folded correctly by forming disulfide bridges. The correctly folded polypeptide chain is then packaged into a vesicle upon leaving the ER in destination for the cis-Golgi apparatus, where it will undergo glycosylation. The glycosyl transferases add 2 phosphates from 2-UDP and 2-glutenax, and then removes the 2-glutenax, leaving a mannose 6-phosphate attached to the protein that then travels to the trans-Golgi. Once in the trans-Golgi, it binds to the M6P receptor, which signals transfer to the endosome via transport vesicles. The Golgi adds specific Rab proteins to the newly formed vesicle that will bind to its target membrane. Once in range, the tethering proteins binds to the Rab protein and starts the docking phase. A V snare on the vesicle and a T snare on the target membrane will bind to each other. They pull the vesicle to its target membrane, where fusion into the membrane will take place. The snare catalyzes the final fusion between the two membranes, allowing the cargo protein inside to be delivered to the outside of the cell.